Now we'll go the further process of fertilization statement, whatever the stages and development of the embryo. At the time of fertilizations will take place in the ampulla, and then what happens is known as cleavage. So what is cleavage? It is serial cell divisions of that part. In previous slide, here we can see this cell is divided into the so many number of the cells, and initially 2 or 3 days this part is protected by the zona pellucida because the lining epithelia of fallopian tubes is having capacity to implant this fertilized ovum, but this is not needed at that time, and that's why it's protected by zona pellucida. In this way the zona pellucida is functions is to prevent the abnormal or ectopic pregnancy in fallopian tubes. And when this serial divisions or continuous division of zygote means process of cleavage is reached up to the 16 cell stage, at that time it looks like the mulberry, or it's a group of the so many grapes like structure is known as morula. This morula is 16 cell stage embryo, and now is gradually reached into the uterine tube approximately third days of pregnancy. Then what happens? It is this fertilizations. We know there is no any nutritional systems. There is no blood supply, and that's why we have also learned one more thing during the oogenesis. It is unequal distribution of cytoplasm. That means more cytoplasms in oocyte is responsible for nutritional purpose also, which is not present in male. That's why this cytoplasm is also provide the nutrition. Apart from this cytoplasms, now in the uterine cavity, now zona pellucida is disturbed or removed, and due to that what happens, it takes the fluid from the surrounding environment. This uterine fluid is now enter into the in between the cells of morula, and in this way the collections of the fluid is known as blastocyst formations. Cyst means collections of fluid. Here it is formations like the collections of fluid. In that way, it also provides nutrition, and in this way, one more things we can see here. The one group of the cells are connected in this pod. It is known as embryonic pods, from which the future baby or the embryo will develop. Another cells which are arranged on the peripheral parts, it is known as trophoblastic layer. Outer cell layer is trophoblastic. Tropho means nutrition. From this, whatever the structures which are held for nutrition or accessory structures to the embryo will develop from this, particularly the placenta, its membranes, and some another structures will develop from this. Other than the embryo, all the structures will develop from this trophoblast. Tropho means nutrition. So here it divides into the two cell layer due to the collections of fluid. What the inner cell mass, outer cell mass. Inner cell mass will develop the embryo proper, and outer cell mass will develop the trophoblast. Now this morula, cells of morula of outer trophoblast inner cell mass is arranged into a one linear fashion, so longitudinally these inner mass of cells are arranged into the two layers. One is above and one is below. The cells which are columnar type of and situated above is known as epiblast, and another is hypoblast. So epiblast and hypoblast cells. These cells are now arranged into a one particular or its role in formation of zones, also known as respectively ectoderms and endoderms. So these endoderm ectoderm cells which are developing from these two layers. So initially the inner cell mass or embryo proper is converted into a two layer cells, epiblast, hypoblast, or also known as ectodermal and endodermal cells. Now what happens in the further differentiations of the cells? Now this fluid is also present around that part, but one more things will take place. This trophoblastic cells which develops the another one structure is known as extra embryonic mesoderms. What? Extra embryonic mesoderms. In this way, this developing embryo along with the trophoblast, now it's surrounded by the extra embryonic mesoderm. As the name suggests, it doesn't take part in formations of any tissues of fetus. That's why it is extra embryonic mesoderms. Simultaneously, the inner cell mass means endodermal cells now line the whole inner surface of trophoblastic cavities, and in this way it's converted into a one fluid cavity is known as primary yolk sac. This is primary yolk sac, and another cells ectodermal cells which again may lining the upper part of embryo, and it is converted in amniotic cavities. Now here we can see this is the embryonic disc-like structures. It is only bilaminar. This bilaminar embryonic disc is surrounded by the two fluid cavities. One is amniotic fluid cavities above and primary yolk sac below, and it's surrounded by the extra embryonic mesoderm. Then further differentiations of embryo blast, and this embryo is what happens. So many initially there are so many small cavities will develop in this extra embryonic mesoderm, but ultimately all these cavities are fused with each other, and in this way this makes the very large cavity. This large cavity surrounding the developing embryo is known as extra embryonic xylem or cavity. Extra embryonic xylem, so large cavity. These cavities extend almost all the extra embryonic mesoderm except the most this part where there is no development of any cavities, means unsplit part of extra embryonic mesoderms. In future it's responsible for development of the umbilical cord is known as connecting stalk. This part is known as connecting stalk. The remaining part of extra embryonic mesoderms is divides into the two layers, outer and inner. The outer is known as somatopleuric layers, and inner one is known as visceral or splanchnopluric layers. In there say it divides into the two layers, somatopleuric and splanchnopluric layer of extra embryonic mesoderm. This somatopleuric cell layers is fused with the trophoblastic cells. In this way, there is fusions of trophoblastic cells along with the somatopleuric layers, and it's converted into a one membrane-like structure. This outer membrane is known as chorion. What it is? Chorion, important membrane during the development of fetus. This parietal layer is also going towards the amniotic cavity and then turn again, and in this way there is amniotic cell layers and parietals or the somatopleuric cell layers of extra embryonic mesoderms. Here it is fused with each other and converted into a one more membrane is known as amnion, and this is secretes the fluid is known as amniotic fluid. Now we can see the embryo is surrounded by the three cavities: amniotic cavity, now this secondary yolk sacs, and outside there is the extra embryonic cylindro. Ultimately this is the floating embryo. Now it is float in the so many fluid cavities. Due to that it's protected by from the surrounding structure. So this is known as the formations of the or further development of the embryo, and now gradually it's converted into the formations of germ layers. Initially it's having the only and only two germ layers, ectoderms and endoderms. Now what happens further we'll discuss. Yes, formations of germ layers, further development. Previously we have discussed there is the embryonic disc is surrounded by the so many fluid cavity. It's having a only two layers, ectoderms or columnar cells and endoderms or cuboidal cells. Now in between these two layers what happens? Two important structures or one process is developed is known as gastrulations. So what is gastrulations? The gastrulations is the important event during which the third germ layers means intra-embryonic mesoderms is developed. In this way, embryo having the all three germs layer is known as gastrulations. How it take place? Approximately this process is take place around the starting of the third week of embryo. Now two cells are differentiated in ectoderms and endoderms. A few cells at the cranial end of the endoderms is proliferated and converted into the one mass of the cells is known as one procaudal plate. It is known as procaudal plate. And another mass of the cells of ectoderms is proliferated at the caudal end of ectoderms. It is known as the primitive strip. Ultimately, according to recent investigations, all the three germ layers are developing from ectoderms. How? So initially there is the two layer cells we have discussed in the previous slides. One is above and one is below ectoderms and endoderm. But now cells of these ectoderms will proliferate and migrate in the deeper part like this, and ultimately is reach up to the endoderm. Now these cells, modified cells from these ectodermal cells is replace the previously placed cells. In this way all the cells of endoderms is replaced by the cells of ectoderms. In this way the first germ layers endoderm is formed by ectoderms. So this is endoderms. Then another cells developing, it is the mesoderms, intra-embryonic mesoderms, cells of the primitive strip which migrates and spread on both layers of the cells, and it's extend from cranial end of primitive strip to the caudal end of procaudal plate. This structure is known as the notochord. In future we will be developed what is notochord. The notochord is the structures derived or in between the primitive stick and procaudal plate. In the regions of procaudal plate, these cuboidal cells or endodermal cells are there, and primitive stick is develops. This primitive stick is actually the formed due to the proliferation of. Now one more development of the cells we'll discuss is the